Thank you for joining us today at this event hosted by LabRoots called Precision Medicine, Genomics, Proteomics, and Molecular Diagnostics Virtual Event Series. My name is Lia Abarzua, and I'm an application scientist at Covaris, a Perkin-Elmer company. Today, I'm excited to walk you through our work on the truCOLLECT system. Our goal with truCOLLECT is simple: to obtain venipuncture-quality results with the simplicity of a fingerstick. By the end of this session, I hope you'll see how valuable this solution is. So this is the agenda that I'll be going over today. truCOLLECT whole blood collection solution is what we'll be starting with first, what it is, how it works, and the problem it's designed to solve in today's genomic landscape. Following that, I'll introduce our truCOVER whole genome sequencing library prep kits, where you can obtain sequence-ready DNA. And lastly, I'll go over a comprehensive workflow from a patient sample collection all the way to NGS-ready DNA. So what does truCOLLECT actually look like? It starts with a patient fingerstick collection. There's no need to schedule an appointment with a trained phlebotomist. When the patient fingersticks themselves, 50 microliters of blood is stabilized on the blood tube and it turns into a desiccated format, which then allows for ambient shipping and storage. When the lab receives the blood tubes, they can then extract and purify. To understand why truCOLLECT exists today, it helps to look at the landscape of genomics. The field is being reshaped by three powerful trends. First is decentralized care. Decentralized clinical trials, remote patient engagement, and growth at home diagnostics is needed. Patients and study participants increasingly expect to engage with healthcare from wherever they are. Next is longitudinal interest. Researchers want to track patients over time through longitudinal population studies, post-treatment monitoring, and repeated sampling. Lastly is scale and efficiency. There's population-wide studies, large cohort enrollments, and cost pressure on labs that are pushing the industry to find smarter and cheaper workflows. So these trends converge on two core requirements for any solution. First, it must be accessible and convenient, so there needs to be a solution to drive participation and adherence. Second, it needs to be scalable and cost efficient, where it expands reach while also reducing cost. The problem is that traditional phlebotomy, the gold standard for blood collection, simply was not designed to meet these demands. Focusing on access and convenience, it requires an in-clinic visit with a trained phlebotomist. When the sample is collected through a venous draw, it's usually painful as well. Additionally, it's logistically complex and expensive to coordinate, especially at scale or across remote geographies. Now, looking at the scalability and cost efficiency, there's limited stability of the collected sample, which in turn means there needs to be cold chain shipping and storage. There's also staffing shortages, which limit the scale, and there's usually manual labeling and handling, which can introduce error. And overall, the workflow is costly and difficult to automate. None of these characteristics align with what modern genomic programs need. Traditional phlebotomy is, in short, a bottleneck, and it's holding back research from reaching its full potential. So despite all these challenges of collection, whole blood remains the gold standard analyte, and for good reason. From a DNA quality perspective, whole blood delivers reduced analyte loss, low microbial contamination, and consistency across patients. In terms of biological fidelity, it preserves critical biomarkers, detects and monitors key biological parameters over time. And lastly, standardization. There's a more standardized, scalable workflow that's compatible for multi-omic applications. So the challenge isn't whether to use whole blood, it's how to access it at a scale. And that's exactly the gap that truCOLLECT fills. So what's the solution? We need a new approach that unlocks whole blood at scale, making it more accessible and convenient, while also having a scalable, cost-efficient, and high-quality extraction workflow. And that's exactly the foundation of the truCOLLECT system. It's a purpose-built solution that delivers the biological quality of venipuncture whole blood without its logistical and procedural burden. So let me walk you through how truCOLLECT works. The truCOLLECT system is a two-component system where it starts with the truCOLLECT whole blood collection kit. The patient or participant finger sticks themselves, and it collects 50 microliters of blood. The blood is then stabilized on device and becomes desiccated. Because of the desiccation, we can then ship with ambient shipping, and when the samples arrive at lab, you can assemble it into up to 96 well format. Now, this is when the truCOLLECT DNA extraction kit begins. The lab user can add the DNA extraction buffer, and then using Covaris' adaptive focused acoustic technology, or AFA, it could perform rehydration, lysis, and extraction from the desiccated blood, followed by a magnetic bead purification on an isopure or similar automatic platform, where then the DNA can be loaded onto an NGS-ready platform or downstream analysis. So let's look at how truCOLLECT maps to the three core pillars that we identified. On access and convenience, it's an at-home, easy finger stick collection of just 50 microliters of blood. The device then enables stabilization for the desiccation of the whole blood. Looking at scalability and cost efficiency, there's ambient temperature shipping due to the desiccation of the blood, which requires no cold chain logistics. Each of the blood tubes comes individually barcoded for removing any mislabeling errors and enables high throughput automation friendly extraction. Additionally, we're able to decrease the workflow cost between 25 and 40%. Lastly, for the last pillar, it's the high quality DNA, where we preserve the biomarkers in DNA quality in the desiccated form. Additionally, this helps sample quality being preserved for extended time before processing. And lastly, we have an optimized workflow where we optimize for high DNA yield. So, how does truCOLLECT compare to other sample collection methods available today? We have the truCOLLECT solution, followed by traditional phlebotomy, saliva-based collection, and dried blood spots. So if you look at the truCOLLECT solution, there's strong advantages across the board for patient-centric factors along with lab-centric factors. In comparison, traditional phlebotomy has strong advantages for analyte usability and sample yield and quality. However, it has limited advantage and moderate advantage across other factors, including sample format, user-friendly collection, geographic reach and scale, and minimal pre-analytical variability. If you take a look at the saliva-based collection, overall, it has a moderate advantage, but really has a limited advantage with minimal pre-analytical variability. And lastly, dried blood spots. It has a strong advantage for logistics and storage, but it really struggles and has limited advantage on analyte usability, sample yield and quality, and minimal pre-analytical variability. So if you take a look at all four of the methods, truCOLLECT is the only solution that achieves strong advantages across all of the categories for both the patient and for the laboratory. So where can truCOLLECT take you? You could use truCOLLECT in a wide variety of different applications, such as longitudinal studies, population studies, pediatric testing, precision medicine testing, and neurogenetic research. So in short, truCOLLECT is a platform solution, applicable wherever you need whole blood quality genomics without the constraints of traditional venipuncture. So I want to walk you through some of the data that we generated with collaboration with Ambry Genetics, a Tempus company. This work was published as a technical note, and as a quick disclosure, this tech note does not indicate or imply that Ambry has adopted this device for clinical testing. So the key features of the truCOLLECT system highlighted in this collaboration include decentralized microsampling, active desiccation technology, and adaptive focused acoustic extraction, or AFA, on the R230 focused ultrasonicator platform for seamless, hands-free DNA isolation. Additionally, you also get high molecular weight DNA recovery, where the high quality DNA is from just 50 microliters of the blood. And lastly, there's a universal assay compatibility. You could use it with any standard NGS pipeline assays and standard orthogonal clinical assays. Following this is the benefits and resulting outcomes for laboratories. You can expand access, eliminates cold chain logistics, it accelerates lab throughput because you're able to do this in up to a 96-well format, and it maximizes genomic fidelity. And lastly, it also drives operational profitability for the lab. So let's look at the extraction parameters side by side. First, we have the dry stabilized blood, which is collected by truCOLLECT. Then you have EDTA blood from phlebotomy. And lastly, you have saliva, which was self-collected. Right off the bat, you could see that the truCOLLECT requires a very small input amount of 50 microliters in comparison to the EDTA blood, which requires 400 microliters, or the saliva, which requires even more at 480 microliters. Additionally, the extraction methods varied. So with the dry stabilized blood, which is truCOLLECT, Ambry used the truCOLLECT DNA extraction kit, followed by the isopure-mini. For the EDTA blood and saliva, they used the MagMax Multi-Sample Ultra 2.0 plus the Kingfisher Presto. Now, something that's worth noting is the shorter processing time with truCOLLECT using the truCOLLECT DNA extraction kit and the isopure-mini. This whole workflow was 2 hours, saving you or the lab another half hour of work because the EDTA blood and saliva has a 2.5-hour extraction time. So for the downstream analysis and QC conducted in collaboration with Ambry Genetics, we evaluated the truCOLLECT samples across a comprehensive set of assays. For pre-sequencing, we looked at QC concentration, yield, and purity assessment. Ambry Genetics also has a clinical cancer NGS panel, which we also ran the truCOLLECT samples on, and we looked at generic metrics such as mean, coverage, and uniformity for sequencing. Lastly, we did a variant concordance with truCOLLECT samples run against known matched clinical samples. We wanted to see how truCOLLECT samples would work with orthogonal clinical testing, such as Sanger sequencing and MLPA. So let's take a look at the results. So from just 15 microliters of blood, all 32 samples had great yield, averaging 1,399 nanograms of total DNA at a concentration of 15.1 nanograms per microliter. The purity metrics fell within the standard parameters, meaning there was enough high-quality starting material for downstream genomic work. To the left, you can see that there's a dot plot, which shows the mean coverage by specimen type: blood, buccal, Covaris positive, which is the truCOLLECT samples, and saliva. The truCOLLECT samples had a 372x mean coverage, which is comparable to both the historical clinical blood and buccal swab mean coverage. As you can see, saliva showed more variability in a lower mean coverage, which reflects its known pre-analytical challenges. Now, because we are working with just 50 microliters in comparison to the 400 microliters of blood, truCOLLECT samples did show a slightly higher PCR duplication rate at 0.32 compared to the 0.26 for blood. But the main key difference takeaway is the difference had zero measurable impact on variant calling accuracy, specificity, or assay uniformity. And the most compelling point is none of those truCOLLECT samples failed sequencing pipeline QC. In comparison, saliva had a 27.77 pipeline failure rate. So this slide shows the concordance analysis against Ambry Genetics clinical cancer NGS panel. So when we ran the NGS cancer panel against the known positives, truCOLLECT identified 1,969 calls of the 1,971 calls, which gives us a concordance of 99.9% compared to traditional liquid EDTA blood. Now for Sanger sequencing, we also wanted to see how the truCOLLECT DNA held up against orthogonal workflows. So for Sanger sequencing, which provides an independent confirmation of NGS called variants at the nucleotide level, we ran across 5 different donors with confirmed variants in significant genes. Every variant was confirmed from truCOLLECT in comparison to the matched DNA, in comparison to the matched clinical samples. So if you look at the trace scores and the continuous long read length scores, you can see that the truCOLLECT values and the original clinical sequencing values are equivalent. So these results confirm that truCOLLECT-derived DNA supports high-quality Sanger sequencing assays, which is a critical orthogonal assay for confirmation workflows. Next, we ran the multiplex ligation-dependent probe amplification assay, another important orthogonal assay used to detect large-scale copy number variants, such as exon deletions and duplications. This is a more technically demanding assay, so it provides a rigorous test of DNA. As you can see across the 6 donors, all of the calls were confirmed. This is 100% concordance across all calls from the clinical known samples to the truCOLLECT samples. If you also take a look at the COVER quality metrics, they're equivalent between the truCOLLECT and clinical samples. So this data demonstrates that truCOLLECT-derived DNA is fully suitable for MLPA-based testing. And finally, we evaluated truCOLLECT using a targeted array platform, which assesses the copy number variants across the 8 donors with alterations in the genes. Similarly to other assays, you can see that all of the truCOLLECT samples had achieved 100% variant detection with the clinical known samples. So in summary, this 100% detection rate across a targeted array platform rounds out a very comprehensive validation dataset. Starting from Sanger, MLPA assays, and variant detection, it's strong evidence that truCOLLECT performs equivalently to venipuncture blood across multiple orthogonal assay modalities. So in short, truCOLLECT is genuinely integrated solution. It's from self-collection and active desiccation stabilization all the way through AFA-powered extraction and seamless NGS pipeline integration. So it's built to help labs scale with improved efficiency, cost-effectiveness, and delivers high-quality data without any compromise. So I covered the truCOLLECT collection device kit and the extraction kit of the truCOLLECT solution, and now I'm going to introduce another component of Covaris's comprehensive genomic solution, the truCOVER whole genome sequencing library prep kit. The truCOVER library prep kit is built on the same foundation of Covaris's precision adaptive focused acoustic technology, and it's designed to deliver true efficiency and true performance for whole genome sequencing library preparation. So let me walk you through the benefits of truCOVER. The truCOVER is actually composed of 2 different kits. You have the truCOVER whole genome sequencing PCR-free library prep kit, which is designed for high-quality whole genome sequencing applications. And we also have a truCOVER library amplification kit, which extends the workflow to lower input samples by adding a controlled amplification step while maintaining high library complexity and variant calling accuracy. So together, these kits give the labs the flexibility to handle a range of sample inputs and application requirements, all within a standardized automation-ready workflow. So let me walk you through the advantages of using truCOVER. First off, it's a high variant calling accuracy. You get standard variant calling with input as low as 0.1 nanograms. Additionally, for SNPs and indels, you get consistently lower false negative or positive rates. And in comparison to competitor workflows, you're able to achieve a 2 to 6% improved F1 variant calling. Next, we're going to look at uniform unbiased coverage. Minimized GC bias gives more uniform coverage in comparison to enzymes, and with minimal amplification-induced bias because of the robust coverage across broad input. Lastly, you have uniform insert size, which improves variant calling. Now, taking a look at the higher coverage and complexity, you get 2x higher library conversion in comparison with a competitor with the same input DNA. Also, for the same input DNA versus a competitor, you get a 30 to 50% greater library complexity for the same input amount, and you consistently get a higher depth of coverage. Looking at efficiency and scalable workflow, you get a 30% faster turnaround time in comparison to key competitors, and you can pool by mass or volume. So there's no need of an additional qPCR quantification. Lastly, the standardized workflow produces reproducible results. So if you take a look also at the robust performance for low input in all sample types, you get high quality DNA from even 1 nanogram of FFPE DNA or zero point 0.1 nanograms of high quality DNA, reducing the QNS rate. Lastly, we see a linear amplification across input types from zero point 0.1 to 10 nanograms, and we're compatible with all sample types: blood, saliva, FFPE, cell culture, and microbial DNA, to name a few. So we've now covered the truCOLLECT system, followed by the truCOVER library prep kit. And in this final section, I'll show you how these two components come together into a truly comprehensive end-to-end workflow solution. This is where the power of Covaris' integrated platform becomes clear. From the patient's fingertip all the way to having sequence-ready DNA in a unified, validated, automation-friendly workflow. So let me frame the larger challenge that this comprehensive workflow is designed to solve. On the extraction side, the traditional challenges are well known. Whole blood requires a phlebotomist, the storage and shipment of the blood tubes is costly, as well as the remote collection device, and the consistency of DNA quality is needed to streamline multiple applications. Taking a look at the library prep challenges, usually it's a very complex and manual workflow, which increases the hands-on time and also introducing potential errors. You usually get suboptimal sequencing performance from saliva and dried blood spots, which usually means that sequencing has to be redone due to the failure, which could also add to costs. So there's a critical need to having a solution that is scalable and batch size and has batch size flexibility. It's easy to implement with automation for operational efficiency, and we're looking to ensure high quality data from a reproducible and reliable performance workflow. And lastly, a simple finger stick approach would be ideal. So here's the fully integrated Covaris workflow from blood collection to sequence-ready DNA. It begins with the truCOLLECT collection kit, followed by the truCOLLECT accessory kit for accessioning. And when the samples are arranged in a 96 ball format, then the truCOLLECT extraction kit can be done, the rehydration, extraction, and lysis of the desiccated blood, followed by using truCOVER DNA library prep to have the DNA ready for sequencing. Let me walk you through some numbers. So starting with the traditional phlebotomy workflow, the blood collection happens at a clinic with a trained phlebotomist. Because the sample is not stable, it needs cold chain storage and shipping. And when it's accessioned, when it arrives at the lab, it usually requires aliquoting before extraction and purification before library prep. That whole workflow is usually $85 to $120 per sample. Now, if you look at the truCOLLECT and truCOVER workflow, you're able to save between 25 and 40% of the workflow costs because using the blood collection with truCOLLECT, it is desiccated and stabilized on device, which gets rid of the cost cold chain logistics, and you could do ambient shipping. When the samples arrive on lab, no aliquoting is necessary, and the rehydration and extraction happens with the same tubes that they were accessioned in. After purification, you could use truCOVER to shear and do the library prep. So this whole workflow of truCOLLECT plus truCOVER ends up being $64 to $66 per sample, which is about a $23 to $56 savings. So if a lab is working on 10,000 samples, the lab could potentially save between $200,000 and $600,000 in workflow costs. Additionally, truCOLLECT has shown equivalent or better performance on uniformity, mean coverage, and duplication rate to EDTA blood, which I'll go over in the following slides. Let me share another important data set. This one is from a webinar presented by the University of Florida College of Medicine and the University of Puerto Rico Medical Sciences Campus in collaboration with Covaris. So the goal of this was to evaluate truCOLLECT as an alternative to traditional venous blood draws for DNA extraction and whole genome sequencing, with a specific focus on pediatric and autism research. Another goal of this study was to have a broader aim of making population-based genomics more inclusive and less burdensome for the participants. So for this study, the pediatric participants and its family members enrolled in this decentralized clinic in Puerto Rico, where a trained phlebotomist got both venous and finger stip samples, so we had matched sampling. Additionally, the venous blood samples required cold chain handling, including freezing on dry ice prior to DNA extraction. In comparison, the truCOLLECT samples were stabilized at room temperature for up to 6 months and shipped by standard mail, which significantly simplified logistics and cost savings. So taking a look at the data collected from truCOLLECT and truCOVER from the truCOLLECT samples and venipuncture samples, the truCOLLECT samples had an average library yield of 94.3 nanograms, while the venipuncture samples had an average yield of 72.1 nanograms. For the average library fragment size for the truCOLLECT samples, it had an average base pair size of 751, while the venipuncture samples had 681. So the percent read maps to the genome were equivalent and consistently high, above the 98% for both truCOLLECT and venipuncture samples. The GC coverage showed comparable uniformity between the truCOLLECT and venipuncture. If you also look at the PharmaCat panel, it showed 100% concordance between the libraries and the truCOLLECT and venipuncture samples. The libraries from truCOLLECT showed greater coverage of around 25x across gene targets spanning GC contents of 32 to 68%. So this real-world decentralized study confirms that truCOLLECT can be used for demanding pediatric and neurogenetic genomics research. Beyond sequencing performance, one of the most operationally valuable properties of truCOLLECT is the sample stability. We were able to achieve reliable and consistent recovery from samples stored at room temperature from 3 to 6 months. Additionally, the ambient stability minimizes the pre-analytical variability, which is common to other sample collection methods, such as venipuncture or saliva. DNA quantity and quality remained consistent for multiple donors after storing the sample at room temperature for 3 months. The DNA quantity correlated with the white blood cell count, which confirms biological fidelity. Lastly, the samples maintained stability when exposed to high temperatures for 5 hours. There were 4 different temperatures: room temperature, 50 degrees Celsius, 65 degrees Celsius, and 75 degrees Celsius. So the proprietary matrix preserved the nucleic acids effectively despite the thermal stress. So as you can see, this level of stability is critical for real-world deployments. Samples were collected in a diverse climate. They could be shipped by standard mail or held in non-controlled environments before lab processing, which will perform reliably with truCOLLECT, as shown from this data. So how do you get started with the truCOLLECT solution today? The truCOLLECT whole blood collection kit comes in packs of 10 or 100. Everything that is needed for the participant to self-collect is included with this kit, and it comes with instructions for use. There's also the truCOLLECT whole blood extraction kit, which can process up to 96 samples, and again has all of the consumables and reagents needed to do this extraction. This solution is listed with FDA as a Class 1 device, and the truCOLLECT solution is powered by adaptive focused acoustic technology and is run on our R230 focused ultrasonicator. I want to say thank you so much for your time and attention today, and I'm excited to help you. If you have any questions, please reach out to us at our website or via email. Thank you so much.